Hello, my name is Sai, and today we are going over polymerase chain reaction for the Big Bang BENG 168 video series. It can be extremely difficult to deal with extremely small samples of DNA. To counteract this, scientists use techniques such as PCR, or polymerase chain reaction, to amplify a target sequence of DNA they have by up to 1,000,000-fold. There are 4 essential ingredients to complete PCR. The first is 2 synthetic oligonucleotide primers. These are primers complementary to regions on opposite strands that flank the target DNA sequence and bind to cut the DNA's sticky ends. The next ingredient is a template sequence of the DNA sample that lies between primer binding sites, and it can be anywhere from 100 to 35,000 base pairs in length. The next ingredient is an enzyme known as DNA polymerase that is used in DNA replication to attach nucleotides to template strands of DNA with the corresponding nucleotide. The DNA polymerase must also be stable when heated to temperatures up to 95 degrees Celsius. The next ingredients are the 4 deoxyribonucleotides that contain the 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. The final ingredient is the pH neutralizing buffer that keeps the entire reaction at the proper pH throughout each cycle to make sure that the DNA polymerase isn't denatured. The first step of polymerase chain reaction is the denaturation of DNA. In this step, all the ingredients, especially the DNA sample, is raised to a temperature of 95 degrees Celsius to split the DNA sample into its 2 strands. The next step is renaturation, where the temperature is lowered to 55 to 65 degrees Celsius and the primers attach to the ends of the DNA at their complementary sequences. In the final step, extension occurs at 70 degrees Celsius, where the DNA polymerase uses the nucleotides to rebuild the strands starting from the 3-hydroxyl end of each primer. These extend past the necessary part of the DNA and are known as long templates. These steps are then run again. In the second cycle, the original DNA strands and the long templates are denatured, and the second cycle results in short templates. These are DNA strands that have a primer sequence at one end and the sequence complementary to its other primer at its other end. These are the strands that are the goal of PCR, and by this 30th cycle, the short templates dominate, having 1,000,000 more times copies than the long templates, or the original DNA. Scientists may also need amplifications for entire cDNA strands rather than specific sequences. cDNA is DNA that has been transcribed from mRNA using the enzyme reverse transcriptase. To complete this, an oligonucleotide that consists of an oligo DT sequence followed by a PCR primer sequence at the 5' end must be added to a purified poly-A mRNA preparation. Using the enzyme reverse transcriptase, the mRNA is turned into a cDNA molecule. The oligonucleotide and the primer then pair to the end of the cDNA, which prevents tandem repeats from forming at the 5 ends of the full-length cDNA strands. Then the traditional steps of PCR occur, and full-length double-stranded cDNA molecules are created. PCR is also handy for gene synthesis. For this, you start with 2 overlapping oligonucleotides, A and B in this case, that then get filled in to make a DNA molecule. Then this strand is denatured and oligonucleotides C and D are overlapped with each strand, C with one strand and D with another. Then this strand is renatured and filled in to make a DNA molecule, one with segments A, B, and C, and one with segments A, B, and D. The A, B, and C and A, B, D strands are then denatured and their strands are mixed together to overlap on the A, B sections, creating 2 CABD strands. These strands are then renatured and filled in, and this can continue for oligonucleotides E and F until the gene is complete. Our source for this video is Molecular Biotechnology: Principles and Applications of Recombinant DNA, 4th edition by Bernard Glick et al.